it's also my great pleasure and to be here and to join this symposium. The only thing I feel regret is that it's such a nice day. We have to stay inside, and so I hope I can stay longer and to do some sightseeing after this. So as you heard from Charles, the human genomic information carries decide who we are. But those information actually traditionally are only presented in a kind of one linear so-called dimensions. But in fact, we know that the human genome, like anything else, they're existing in the nuclear space, but in fact, they're all kind of organized within a three-dimensional manner. And so to really understand how the genome works, we have to understand how those players, that including DNA, protein, and RNAs, how they kind of organize together within the defined limited space. So collectively, we now kind of describe this so-called space as a 3D and nucleon. Well, why we care about this three dimensions, and in more particular within to understand the genome is that we think that the three-dimensional organization matters. Not only they're organized in a chromosome and the nearby so-called the neighborhood, but they are also dynamic in space and time. And they're also related to the disease, and many of those are involved in the genome stability and the instability. They're all kind of related to how the genomes are organized. And we also found that there are lots of the large majority of so-called so far identified genetic variants are in fact they are located in the non-encoding regions. So how those kind of information could really affect how the genome is organized is really a key question that we're trying to address. So now the question is how to study the three-dimensional or even the four-dimension, that is the 3D plus time. So to simplify this and hope to describe those technologies that can be applied to study such questions in terms of nuclear imaging technologies, such as using the microscope. And we also, because of the recent genome advance, and we also developed the genome mapping technology that in fact can map the coordinates of how the genome are kind of interacting with each other in the XYZ dimensions. And of course, with all those data that we gather together, whether it's by the imaging or mapping methodologies, we all have to using a computational approach to put the things together and try to understand what they mean to us. So first, the microscope views using the advanced DNA fish as well as super resolutions or confocal microscopies would be able actually to visualize how exactly and the chromosomes or the genomes are organized existing within the nucleus at a particular time of moment. However, the problem is that the imaging-based approach and there are lower resolutions, so you cannot really relate the structures that you see at the nucleotide levels, so therefore you cannot really relate such structures to specific genes or functions. So in 2009, my group and along with the other group, so we developed the 3D genome mapping technology. And the method that we developed is called the CHIAPAD. It stands for Chromatin Interaction Analysis by Pattern Sequencing. So another method actually called is high dimension chromosome capture sequencing. So both approaches actually share the same technical concept, which is that you can use formaldehyde or chemical cross-linking to fix a particular moment of the nuclear state. Now, after that, you can use molecular manipulations to generate the chromatin fragments, and then conduct the nuclear proximity ligations to establish artificial so-called connectivity informations. So after you establish such the linear relationship by ligations, and then we can use either enzymatic approach or physical sharing approach to generate this paired end fragment, and then by using the high throughput sequencing approach to map the two DNA fragments that are linearly located far away within the genome, but spatially they are in close proximity. So using this approach, and if you can generate lots of those informations, and actually we can predict the exactly three-dimensional coordinates of the DNA fragment where they're located in the XYZ situations. So in terms of the ChIA-PAD method specifically, it does not only generate those ligation informations, but it also at the same time capture the specific protein binding information, because we're using the specific antibody to pull down a particular protein factors. So in this way, this approach becomes more specific, and the information that is derived from the sequencing can actually carry the functional meanings from the information. So in short, this method can generate the transcription factor bindings and the chromatin interactions between the binding site, as well as those interactions of chromatin contact that are randomly taking place within the genome. So that information actually can help us to assess the relative relationship within the nuclear space. So recently, we actually conducted very detailed comparisons between the high-seq experiment, which was generated by using Billings reads from the same DNA fragment, from the same cell lines, and with the ChIA-PAD data that we generated from. So we actually can do the very cross the technology comparisons, and showing that both methods can actually reproducibly capture the higher order chromatin contact information. And the only difference is that the ChIA-PAD method can actually capture very detailed protein factor modified or mediated chromatin interactions between the specific binding sites. So therefore, in conclusions, we see that ChIA-PAD is an ideal method for 3D genome mappings. It is because they're more inclusive, and they produce the multifaceted data that you can predict very refined structure as well as high-order chromatin interactions. And more specifically, it provides the high specificity with the haplotype specificity and the nucleotide resolution, which I will give you more details later on. So what we learned since we developed the 3D genome mapping technology in 2009? So over the 7 years, and the field have come to some conclusions to understand that from the nuclear structures to the chromosome territories and to a very specific so-called topological associated domain as a fundamental structure unit that organizes within the genome, to the more specific how exactly the genes and the promoter and the enhancers, how they interplay with each other to really kind of execute their functions in a regulatory manner. So those are the current questions that we're addressing. So what defines the topological associated domains, and what are the sub-TAD structures, and how the genes are behaved within the TAD structures, and so on and so forth. So our strategy to address those questions and to be with a high precision is to using the ChIA-pad approach with a specific focus on the CTCF, which is a chromatin organization protein, as well as the RNA-POL2, which is a transcription factor that are involved in all transcription activities. And then we're using all the existing epigenomic and the transcriptomic data to annotate such structures. So using this approach, we're starting to generate some very detailed, high accuracy data that we can allow us to really pick into the details how the genomes are organized. So first, by using the CTCF ChIA-pad, we'll be able to identify how the chromatin structures is organized by CTCF. To be sure is that we found that the majority of the CTCF defined chromatin interactions, they're actually with the binding motif facing to each other. And we also identified the binding motif could be organized within the tandem, either facing left or right. And we further found that cohesin, which is another chromatin organization proteins, are highly associated with CTCF, and together they also showed the orientation specificity. And based on the CTCF-defined structures, and we can also, based on the connectivity and the strengths, we can actually further characterize the so-called CTCF-mediated chromatin contact domains. In short, we call it CCD structures. And again, most of the boundary CTCF motif that they're pointing inward. And then we also see that usually the boundary of the CCD structures, it has multiple binding sites and facing toward each other. So that probably provide kind of a double knot, so-called structures, to make sure the boundary is very tightly organized. And this CCD structure, in fact, is highly kind of associated, kind of similar with the highly identified top structures. So in a way that, as you can see, that it doesn't matter what the method that you use, and in consistencies, and we can generate a very similar high-order structures. But the difference is really the details. So what is the details that we see? Now I'll give you a particular example of the CCD structures. In this particular example, it involves 8 CTCF binding sites, and most of the binding side on the boundaries, they're facing to each other, but at the binding side within the CCD structures, they're actually all tandem facing in one direction. So that actually provides some very clear so-called structure preference, how CTCF is involved to organize the structures. And so we said we actually, based on the binding and the looping preference, now we can actually start to realize that the CTCF organized loops actually has two types. One is the helping type of structures if the motifs that converging to each other. And where the other one is more kind of a coiled structures, and if the two motifs are in tandem orientations. So based on such information, it can guide us to start to visualize that how from the two-dimensional informations come down to the three-dimensional informations. In fact, all this 8 CTCF binding side, they're all interconnected together, so that will kind of make us to speculate that and those binding sites are actually, they're all contacted together to form one so-called CTCF foci structures. They probably form a kind of a platform to organize the CTCF in the three-dimensional so-called topological organizations. And we also using the RNA-POR2 to do the chia pad analysis. Previously, that was published in 2012, showing that RNA-POR2 mediated chromatin interactions connects enhancer to promoter, and more specifically, actually, we see many of the promoters of different genes that are actually coming together to form the so-called multi-gene chromatin complexes, which in fact they provide the structure basis for co-transcription of different genes. Linearly, they are passed in different locations, but structurally, they're actually coming together in one particular nuclear space. So now, by putting the CTCF chia pad data and the RNA-POR2 chia pad data together, now you can see that actually we say that most of the RNA-POR2 activities were actually taking place in the CTCF looping anchor regions. And more specifically, this is very interesting, is that compared to the tandem structures with the tandem structure with converging ones, we see most of the transcriptional activities that including RNA-POR2 activity, even the presence of gene promoters, they're most all kind of concentrated within the CTCF anchor region, particularly in the tandem CTCF orientation structures. So that actually suggests that the CTCF boundary has binding has two functions. One is to define the so-called overall domain structures, which has the tandem orientations. What we see in the CTCF structures, and usually they're associated with this tandem coiled structures, which is structurally probably more relaxed to be associated with gene transcriptional activities. And so based on that observations, we can actually propose the working so-called hypothesis, how within the genome, within the nucleus, how CTCF and the RNA-POR2 to working together to shape the chromatin structures, also at the same time provide the topological framework for transcriptions. So here is examples of a two-dimensional CTCF structures versus the RNA-POR2 mediated chromatin structures within the content of genes and of gene promoters and the enhancers. So if we just imagine within the nucleus, only consider the CTCF structures, and we see that the anchor genes or anchor proteins that actually bring together and by the CTCF to form this so-called anchor and also the loop structures. However, if you consider the RNA-POR2 working together with these structures, now you can see that RNA-POR2 through the POR2-mediated chromatin interactions that will bring the loop genes or loop enhancers to the anchor regions structured by the CTCF foci. Very interesting is that we also did the gene expression breadth analysis, means that we take all the expression data, like 60 different tissue data. We found that most of the anchor genes compared to the loop genes, they are more of a constituent, they're less, they're more of the less gene-specific expressions. But further, when you look at the active anchor genes within the particular cell lines, and we can see that most of the anchor genes that in fact the anchor genes that are mostly so-called commonly expressed genes. So, and the last point I want to address would be that how to use this approach to map the haplotype-specific computing interactions. And this shows is that based on fixed genome informations in these particular locations, the father copy has a C, where the mother copy has a G nucleotide. So as you can see that because based on this SharePad mappings, because it is the single nucleotide resolutions, so we can see that the mother copy in fact has very strong CT self-bindings, while the father copies has almost none, just the background CT self information. As you can see that the CT self-mediated computing interaction only existing in the father copy, but not in the mother copy, but not in the other the father copy. So this actually provides a haplotype-specific and the single nucleotide resolution of the mapping of the computing interactions. So with this information, actually we found that statistically most of the CT self-binding properties that are all associated with the canonical CT self-binding motif, while the CT self-loci that they lose or have weak CT self-bindings, they usually have the altered motif structures. In this particular example, we found that, in fact, it is a high risk. A GWAS loci has been identified that are associated with asthma and also several autoimmune disease. So to summarize that, right now we're actually developing a systems approach using the 3D bindings to characterize the genome structures, and also come down to the specific haplotype computing informations, and we can actually categorize to interrogate what are the genes and what are the copies within the human genome that really have the active functions versus they do not have the functions. So in that way, that can actually link to the specific biochemical approach, biochemical functions at the genome structure levels, and to the phenotypical trace, as well as lead to the disease. So at the end, I just want to thank my colleagues because it really involves a wide, broad range of skills that including molecular biology, genetics, genomics, as well as computationals, and also involved many of collaborations from within the institute and also the other part of the world. And also thanking my funding agencies. Thank you very much for your time.